Okay, Mike, we're recording. All our lines, I think, are now tied together. So let us commence. Good afternoon, good morning to you, wherever you may be. This is the next in our astrobiology webinar series. We've just got a few announcements before I pass over to Aaron and Greg. If any of you have been looking for the videos, the recorded videos of the last few activities, they will be posted very shortly. We just had a slight delay on that, so we should be up to date in the next couple of days. Tomorrow's webinar, which was on icy bodies, has had to be postponed, unfortunately. They're just due to some diary clashes of the presenters, so that will be rescheduled and you'll see it appear in the events list. The slides for today's presentation are available again. If you go into the event for today, you'll see a link to those slides. Previously, we've been posting the slides as Google Docs. In this case, today's slides had some moderately complicated pictures in them, and it turned out that Google didn't seem to be rendering those correctly. So there's a PDF file which you can download, which has all of the slides. And then finally, just to remind you, this meeting's being recorded, not just what you say, but also everything that's typed on the screen. So be aware of that. And with that, I will pass over to Aaron and Greg.
Great, thank you. So today we're going to be talking about, well, presenting the white paper for astrobiology features that I think the link just says Last Universal Common Ancestor, but the title is, What Were the Genomic, Metabolic, and Ecological Features at the Root of the Tree of Life? And we're starting with this slide because I think it's always worth pointing out, even though it's obvious to most people these days, that when we're talking about the origin and early evolution of life, we're talking about a process that had many stages, and it's likely that the sorts of questions we're posing today will really only allow us to look at this particular stage, early cellular life, and perhaps the transition towards cellular life. The root of the tree of life, well, before I get into it, I should say, I think that we have a small enough group that Greg and I are just going to give some intro slides, and then we'll go through the questions posed by the white paper, and I think these can be more discussions because there are only 8 of us in the room, and so I don't think we'll be talking over one another. Anyway, so the root of the tree of life, at best, represents a single lineage that we can characterize. At worst, at least, represents the organismal coalescence point of life on Earth. And, we don't yet know all of the characteristics of this organism or population of organisms, and we also don't necessarily know its relation to the origin of life. Okay, but we were considering that it's probably likely that this stage represents an early stage toward organismal individuality that allowed the tree to begin branching, and that perhaps in order to evolve a complex ecosystem, like the sorts of things we're looking for in the astrobiology field taken together, that perhaps this stage, represented by the last universal common ancestor, is a prerequisite for a more complex ecology to evolve. And so it's important not only for understanding the early evolution of life on Earth, but perhaps for understanding the sorts of selection pressures and evolutionary dynamics that would be important for understanding complex ecologies anywhere in the universe. So typically what's been done so far is to take a census of the genomes or other sorts of characteristics across a reasonably representative group on the tree of life. And clearly this sort of approach is becoming more and more sophisticated as we have more and more genomes, and the sorts of database searches that one can do to identify common traits across the tree are more and more sophisticated as well. But there's a second stage in this process that requires extrapolating from these data what would be present in the last universal common ancestor, and that requires an understanding of the evolutionary processes that can produce artifacts in the original data set, as well as more sophisticated phylogenetic approaches. And so this is another area where the tools available have gotten significantly better. And so we think that now is a very good time to be asking the sorts of questions that we pose in the white paper and that we'll be discussing today.
Right. So there are two major approaches to investigating the nature of LUCA, or the nature of microbes that were existing at the time of LUCA, and these are top-down approaches, which Aaron just briefly hit on, which is doing comparative genomics and trying to work backwards to the root of the tree of life, given what we know about existing biological systems. Or you can do bottom-up, and that's the idea of starting from a set of first principles and trying to infer what the environment would be like at the time of the origin of life, what prebiotic chemical activities could have been going on, or even at a more abstract level, ideas of complexity theory of sort of how do you get complex systems that can eventually evolve and become something that we would call life. So the interesting aspect, which you can see right here in the figure as featured by the Earth, is if these two approaches converge, if we have the ability to make inferences from the bottom up and inferences from the top down, and they both produce arrows that point to a similar origin narrative, then that's really encouraging, and we might be on the right track. And developing tools for top-down, therefore, becomes very important because bottom-up has been done for a lot longer, and in some ways, the approaches need to catch up so that they can potentially converge. And just one other thing is off to the right, you can see Mars over there with a different geological history. And of course, we don't have any top-down on Mars, but what's interesting about this approach is it can be generalized. So you can start to make the distinction in origins between what circumstances may have led to the origin of life on Earth versus what hypothetical conditions could lead to the origin of life on exoplanets. And another part of this is the caution we have to use in top-down approaches because of the universal reality of extinction and of evolutionary processes overriding things that may have happened before, either within the genomes themselves or in eliminating other groups of organisms through niche exclusion. And so when we talk about these very early states, we have to be aware of the possibility that when you work backwards and infer, you might be inferring a state which simply replaced a previous living state that we do have no record of. And so you can see on the left, I have a schema here that demonstrates how this may be true of something like an RNA world. If it was completely superseded by biological systems that evolved out of it, there may not actually be any top-down evidence left. Or there may be top-down evidence in the form of cofactors, in the form of ribozymes, and these sorts of things, but that have been co-opted in a different way. So a lot of flexibility is required, really, to integrate these narratives when you get down to origin stories. And the example on the right is just one from history, where we have a lot of ancient texts and books where you can clearly see they've been overwritten, and you can look and see what was written underneath them, and the relationship between those two can actually tell you as much about the history of the book itself as what was written in it. And the other really salient feature of doing reconstruction of LUCA is trying to understand what it is, in fact, you're reconstructing. And is LUCA a cellular organism? Is it no different than a particular microbe or a microbial lineage we could isolate today, which would maybe be the LUCA late scenario, where you have an origin of life, or maybe a pre-Darwinian type of pre-life that existed some time before LUCA. And LUCA was just one of many distinct cellular lineages that just happened to be the ancestor of lineage that exists today. As opposed to a feature on the right, where LUCA actually is a pre-Darwinian, pre-genote state, much more closer to what we would call the origin of life itself. And the major lineages that we observe, bacteria or archaea or even eukaryotes, would have independently diverged from this pre-Darwinian state. And the fact is, we don't know which of these narratives is correct. So hopefully we can use these top-down comparative genomics tools to infer which one of these may be the more correct narrative, or we can see in what ways the approaches we're using are consistent with the inferences that we could make in either case. And finally, another tool that I wanted to briefly mention is ancestral sequence reconstruction. So this is a little bit different than just reconstructing phylogenies of proteins, because you also reconstruct the actual protein at each internal node within the phylogenetic tree. So, for example, if I have an alignment, you can see on the left, and a phylogeny, using some kind of evolutionary model, I can infer the probability of each of those amino acids being at each site of some ancestral protein. And if we can recreate these protein sequences very down to the root of the tree of life and actually synthesize them, then it might be able to tell us an insight into the very earliest biochemistry and the very earliest metabolisms of life and what it was doing, even if those functions are very different from the descendant sequences. And so that's another really exciting area of study that's only starting to be fully explored. So as I touched on very briefly, and what this first question really illustrates is does LUCA represent a single organismal lineage, whether or not that was the first or simply the survivor of some ancient extinction event or some other type of reticulation? Or does the root of the tree of life represent a whole community of organisms that are sharing genes and basically evolving cooperatively? And that's an important question that really needs to be resolved to have a firm understanding of origins. And if it is a more communal organism, then all of a sudden the role of horizontal gene transfer becomes a lot more important than maybe it would be otherwise. So if we observe genomes today, it's very clear that horizontal gene transfer is going on between most, or if not all, of major lineages of the tree of life. So that many genes that exist in current genomes do not necessarily have their origins in the origin of that organismal clade. If you apply this principle consistently, it means that a lot of the genes that may reconstruct to LUCA could have existed in multiple organisms throughout an entire ecosystem, and LUCA itself may not have existed as a single physical entity. So the consequences of that also need to be worked out, and what this means for, well, number one, what should we call LUCA? The ecosystem, the collection of genes in some sort of a pangenome, or the actual organism? And number two, is LUCA in itself a distinct point in space and time, or is it spread out over a long period of time due to a coalescent effect? And we really maybe should even have a different word for it.
Okay, so assuming that LUCA represents a sort of change in the type of transmission of genes from a dominantly horizontal regime to a vertical inheritance regime, we thought it would be useful to understand exactly what selection pressures caused this to take place, or whether they had something to do with a change in biochemistry or genome encoding. And so we think that perhaps you could use evolutionary modeling, other sorts of modeling, and when I say evolutionary modeling, the sort of game theoretical modeling used in ecology, other sort of cellular automata-like models, to get at what are the benefits of having the chief mode of inheritance go from a horizontal, a chiefly horizontal regime to a vertical one. Okay, another question is which gene families had evolved by the time of the root of the evolutionary tree? And this is the most sort of straightforward question from a top-down perspective. People have been constructing minimal genomes of the last universal common ancestor for about 10 years, but they vary in their results pretty wildly. And so continuing along this line of research we think would be important, especially as we have more and more genomes and more sophisticated analytical techniques. And furthermore, could we develop techniques to get a sense of what order certain gene families, or at least the functions that they imparted, emerged before the last universal common ancestor? And this sort of analysis, paired with the growth in metabolic databases, can be used to infer which metabolic networks existed at the root of the evolutionary tree. And we can use the sort of more sophisticated systems biology approaches that have been developed recently, like metabolic flux analysis and that sort of thing, to get a sense of how metabolites moved through such possible metabolic networks. And from there, perhaps we can constrain possible biological environments that LUCA may have inhabited, assuming that LUCA represents a single point in space and time.
So one of the hypotheses that is very important in current research in origin of life is the idea of an RNA world, or some earlier regime where a genotype and phenotype were somehow intermeshed, and replicating nucleic acids really formed the core of a living or a pre-living system. But if we assume this, then logic dictates that at some point information became encoded in DNA instead of RNA. So there's not a lot of direct information to infer when this particular event would have occurred, and this is made more complex by the fact that a lot of the DNA replication machinery between the different major domains of life is actually not related to, say, another domain. For example, DNA polymerases between archaea and bacteria are quite different. So if we can develop new sort of top-down approaches for inferring the relative ages of the proteins associated with these, maybe we can actually determine if the transition to a DNA world occurred at some point before LUCA, or maybe LUCA did have an RNA genome and that there were multiple convergent events across lineages to really make use of DNA, and even if that itself was facilitated by horizontal gene transfer.
So assuming that we can faithfully reconstruct some of the gene families that were present in the last universal common ancestor, can we say something about membrane-related proteins and their relationship to cellularity itself? So a lot of research has been done on the prebiotic formation of amphipathic bilayers. But less has been done on which proteins were present in the membranes of early life. But we know, for example, that the signal recognition particle is composed, which is responsible for moving proteins into membranes, was not only present in the last universal common ancestor, but consists of two paralogs that were present in the last universal common ancestor. So we think that this is an area where we could use ancestral genome reconstruction to inform a better understanding of cellularity at the time of the last universal common ancestor and the relationship of the organism to the external environment. Okay, and then also, if we're able to characterize the proteome of the last universal common ancestor and the genome, does that tell us anything about earlier states? And one of the main vehicles for looking from the top down at the likelihood of certain prebiotic chemistry models is whether or not they reflect the cofactors that we see in ancient proteins. And this is something that the relationship between cofactors and ancient metabolic transitions is something that was pointed out in the 70s, but now with the ability to reconstruct ancestral genomes, we have the ability to investigate this in more detail.
So one of the most challenging aspects of studying LUCA is actually determining when, in fact, LUCA existed. This is long before there's anything like a fossil record, and even though there are isotopic and biomarker and other geochemical records that preserve very early life, even within the Archean, these tend to be very sparse and not necessarily informative to trying to reconstruct the state of an early living system, just maybe whether or not life existed. And there are also abiotic processes that could account for some of these. Because of this, really one of our only tools may be a molecular clock. And using a molecular clock or inferring divergence times based on the evolutionary rate of genes and proteins across phylogenetic trees is more and more challenging the earlier you go back in time. And so one of the major future developments that we really need to break this field open is coming up with ways to actually internally calibrate divergence times within phylogenetic trees and within genome sequences. And if we can do this, we can actually start to get at some really interesting questions. In my mind, one of the most important of is, did LUCA exist before or after the late heavy bombardment? Was the origin of life frustrated by these sort of dramatic events very early in life's history and therefore only arose after 3.8 billion years ago? Or was the pre-LHB Earth amenable to the origin and diversification of life? So you would have maybe LUCA could exist as early as 4.3 billion years ago, where you've had a liquid ocean. And maybe LHB was simply an extreme extinction event establishing basically the fact that we only have a few major domains. But in order to get these questions, we really have to figure out how to do very ancient molecular clock timing, and hopefully we can integrate that with what we do have in the geochemical record. And another part of this is how much time actually occurred between the origin of life and LUCA. If we have a very late LUCA, then maybe there is a very elaborate pre-life and pre-LUCA life narrative that maybe we can fill in some parts of. However, if we infer by clocks or by other methods that LUCA existed very early in the history of the Earth, then the amount of time you really have to do an origin and to have pre-LUCA evolution gets compressed. We have no idea what the rate would have been across each of these scenarios, but this does have astrobiological consequences. If life evolved as soon as there was wet water on the surface, and maybe that tells us something about the prevalence of life across other planets. And if it looks like there was an extensive prebiotic chemical stage or an extensive pre-LUCA stage leading up to a relatively late LUCA, then maybe major geochemical and planetary events such as late heaven bombardment add a little bit of contingency to the type of biology that we observe today.
And then I think finally, is there any genomic evidence in LUCA for interplanetary panspermia? And so I don't know whether there is or there isn't, but we could perhaps look at the conserved gene families for some sort of trace of a function that would, or functions that would confer survivability. It may be difficult then to disentangle these from the late heavy bombardment, but perhaps they might also give us a clue to the region of, or the planet or moon of origin as well. And so this is something that at least is not outside of the field of the last universal common ancestor reconstruction. Okay, so I think that's it for our slides, and I guess the paper will be opened up for comments, but if anybody has any additional questions they'd like to add or delete or comment on, we could discuss those here or I guess over the Astrobiology Futures site or Google Drive, over Google Drive.
Yeah, there are many ways that this could be done, but just to clarify, at the moment the paper is in read-only mode. As soon as we're finished here, it will be flipped to comment mode, which, just in case anyone hasn't done that before, it doesn't allow you to edit the paper directly. What it does allow is you can highlight sections and do the equivalent of attaching a post-it note to it. So it's a great way for being able to add comments directly to the paper. If the discussion that you want to have becomes more complex than you can really do through a series of post-its, then we can open up a discussion thread on the site. But at this point, given that we are a select group here, I was going to say anyone who wants to ask a question, please go right ahead. I can see that Eric is typing, but Eric, if it's easier for you to talk, the lines are, I think, unmuted, or if they're not, then Mike, it's *6, isn't it?
How does that work, Andy? Can you hear me?
Oh, yes, I can hear you very well. Hello.
Okay, thank you. Hi, everybody. Yeah, this is faster and less troublesome than typing. So, yeah, very cool. A lot of bases covered. I think about the sort of detailed way Carl Woese argues about the transition between a heavy gene transfer age and a more vertical age. And when he has time to lay that whole argument out, the change in the character of translation is absolutely central to before and after. And it strikes me that, one could write an entire book about what we don't know and would need to know about the change in the character of translation. And I know, that other people in this group, like Lauren, are working on origin of the ribosome. But how integrally do we want to think about the change in the character of translation as being really part and parcel of how we ask this question? Maybe even, it's a narrow window in time than things like the change in the character of the metabolome or the proteome, but it may be sort of functionally more inherent to the character of this transition.
Do you want to take that, Craig, or should
I? Sure, I can take that.
Okay, I can do it too.
So the as far as translation or the evolution or development of translation relates to that shift, I presume the idea is if you have a shared genetic code, then all of a sudden horizontal gene transfer is much more possible, or even if the genetic code is more plastic, then maybe you have an easier time sharing genetic material and all sorts of different contexts. But what interests me is if we look at the proteins involved in translation, we look at the synthetases and even a lot of the biosynthetic pathways, it seems apparent at the time of LUCA that we had more or less a full translational apparatus and more or less complete genetic code. So arguments from a transition involving translation would suggest that the shift from a Darwinian or pre-Darwinian to a Darwinian or a HGT dominated to a vertical system would almost necessarily be a pre-LUCA shift and not something that would discriminate a pre-Darwinian threshold LUCA from vertically inherited lineages derived from there.
Yeah, I think that's a really great point you made about translation, and I wonder whether so one idea that I think you might have been thinking, Eric, and that Greg voiced was the idea that the genetic code, that a common genetic code might have actually been useful to an era of horizontal gene transfer, heavy inheritance, and that now we see organisms sort of encrypting themselves by altering their genetic code slightly. I was thinking about approaching this from more of a ecological modeling perspective, but certainly if you'd like to flesh out a question that we could add, it's definitely an important aspect to the story, both beforehand and I think, well, first of all, leading up to LUCA, and then also before the time of LUCA. I don't know if that's...
I think an important aspect of that question that also touches on what you mentioned, Eric, is maybe the idea that there's some kind of a game theory at work here too, where do the rules change where it becomes less adaptive to share genetic information because everyone already has everything that's useful, or because the world starts to fill up ecologically and competition therefore becomes a stronger constraint than cooperation? And those kind of ecological game theory questions, like Aaron suggested, maybe would be a really important thing to include in this kind of investigation.
That would be great. Yeah, I'm looking at Eric. I'm looking at what you just typed. That would be great.
Yeah. Thank you. I mean, I don't.
Oh, I think I just lost sound connection.
Eric, did we lose you? We had half a sentence and then you were gone. But I see you're typing now. Right, yeah, we could have solved the problems of astrobiology if only the phone code had been longer.
So close.
All right, so we will leave Eric to retrieve his phone, and let me just ask: other thoughts, questions that people want to raise, suggestions that could help the team strengthen the paper, areas that they may not have covered that you could help them in thinking about?
Hey, can everybody hear me?
Yes, we can.
Hey, it's Lindsay. One of the things that the last couple groups presented that was really kind of a neat feature was a section called Things to Work On in the Coming 10 Years. And since that was sort of the whole idea of this roadmap, was to sort of view this as a, what are we looking for in the next 10 years in terms of experiments and that kind of thing. Do you think, do you guys want to brainstorm a little bit about that now, or, think about something, including maybe something in the paper, just something that seemed to be a really neat feature of the last couple papers that we may want to start incorporating into some of the next one. So wondering what your guys' thoughts were on that.
Well, so I think we went into this assuming that all of these questions were things that we'd like to have answered in the next 10 years. And at least what we tried to do was come up with tractable, or questions that seem to now be tractable either in part or completely, and it's just a matter of human, workforce power. But I think, a summary of what could be done in the next 10 years would be a good addition to the paper. I'm open to that.
Do you have thoughts on this? Anything that was on the fringe of what you think might be possible? Just in response to Lindsay's invitation to brainstorm.
Yeah. Well, I think that tying gene families of the last universal common ancestor to other physiological traits like metabolism or cellularity, or the genome and its maintenance and replication, are things that are now possible thanks to tools that are currently under development. So that's what I'm interested. That's the direction that my interests are going in, and I know Greg is sort of approaching this question from a different approach that maybe you'd like to comment on, Greg, but having to do with timing and top-down and bottom-up approaches together and that sort of thing.
Sure, thanks. So as far as what's on the fringe of a 10-year window, or maybe on the fringe even theoretically, is what I briefly commented on of the, determining sort of new techniques and developing chronograms and molecular clocks to infer a more precise timing of these ancient events. If there happens to be particular events preserved within genome evolution where there is a signal that we can learn to extract, then this becomes very tractable, and it's simply a question of developing the algorithms and the proper calibrations and models. However, I suppose there is a chance that information is forever lost. And if that is true, then we might not even realize it's not tractable until we look for it. So it seems to be slightly different than a question of what can we do in 10 years versus 20, and more a question of if it's possible, I think we'll have it within 10. And by then, we'll also probably know whether or not this approach simply, there's just not enough information left to do it. So I don't know if that's optimistic or pessimistic.
Yeah, I would also like to see, because we're looking at, this is a historical science, so it's not, testable in the lab. So I would like to see a way to sort of more accurately evaluate what's working and what's not working, because there's a lot of stuff out there. It would be great to be able to evaluate it quantitatively.
Other thoughts or questions that people want to raise? Were there things in the paper that anyone feels they wish to vehemently object to, or they have a very different perspective on this? Now is a great time to share your thoughts. And just in case anyone isn't clear, you can, if you're muted, you can unmute with *6.
I'm reading what Eric has typed here, and I think that I think that this is the sort of thing that could be modeled precisely with the sort of ecological models people use to look at selection regimes and evolution.
I have another question just appearing on the text box. Actually, yeah. But those are nice to have as well. Okay. If there are no other questions that that we want raised at this moment, or any point. What I'm going to do is, in a moment, we'll flip the document open for commenting, and we'll encourage everyone to go and do that. If, as I say, if the conversations become too complex, then Aaron and Greg can just give us the nod, and we'll open up a discussion thread about that. We did that on the first couple of papers, but in fact, we found most of the time people just want to have the conversations inside the documents themselves. And just to remind you, in case anyone joined late, tomorrow's webinar has been postponed. So, gentlemen, thank you very much. We will go and change the status of the document, and I look forward to reading the revised version. Thank you.
Great. Thank you.
Bye.
Thanks, everyone.